So, good morning, everybody. This is now, we're moving into mononuclear phagocytes. Today's lecture will be specifically on macrophages, and then on Wednesday, we'll discuss dendritic cells. Okay. I've built in, like the last two lectures, some time for questions as we go along, and then we should have time at the end for questions. And I welcome questions, of course, because I think it helps the discussion. And of course, if I miss something or I'm not communicating something in a way that you understand, please feel free to jump in and ask a question, okay? Besides that, you have my email here in case something comes up after a lecture and you had a question or something that I didn't touch upon. So please feel free to reach out to me as well through email. All right, so let's get started. Today is, as I mentioned, monocyte and macrophage ontogeny. This is the picture we have today. In terms of the ontogeny, the developmental lineages of both macrophages and dendritic cells. And a lot of this picture has changed dramatically in the last 10 years. One of the big concepts that has benefited from ontogeny research is the distinction between a dendritic cell and a macrophage. There had been a long time, and we'll talk more about this during the dendritic cell lectures, but there had been a time, and there still is to an extent, where the difference between these two cell types had been difficult to distinguish. And that's because there's large overlaps in their ontogenies. Specifically, there is one particular lineage, which is the monocyte. And the monocyte can become either a macrophage or a dendritic cell. And I'll talk about that to a certain extent, but just, to quickly or briefly just give you an overview. Now what we understand, a major distinction is between the dendritic cell and the monocyte, the dendritic cell and the macrophage, is that many of macrophages are seated in the prenatal state. So you can see, for example, the erythromyeloid progenitors, erythromyeloid meaning they can differentiate into erythrocytes or myeloid cells. And this happens very early, given rise by the yolk sac, then become either macrophages from the yolk sac that seed the tissues, or monocytes that seed the tissues via the liver. And this is a major distinction between the macrophages as we know them today.
We're almost done with.
Let's see, I'm going to mute Danielle. And dendritic cells here, which come from the adult bone marrow. So now you have a very clear distinction between these two that I'll talk about. And as you've seen in my prior lectures, I do like to give historical perspective. The mononuclear phagocyte system previously referred to the reticuloendothelial system. Reticulo because these macrophages seem to form a network in tissues. And they were often found close to the vascular endothelium, which is why it was called the reticuloendothelial system. It changed in the 70s, being referred to as the mononuclear phagocyte system. So from Simon Gordon's perspective here, as well as from Simon Yona et al., sort of more of the recent changes or considerations that have been made in this area. And in terms of mononuclear phagocyte system, this term was coined by Zanville Cohen Van Firth. This name you'll see quite frequently, and Hirsch. Just to sort of give you a overview again of what macrophages, where these macrophages are, a lot of them which have been studied for decades and decades. You have the Langerhans cells. Actually, there's a very interesting story about Langerhans cells because they were initially the quintessential dendritic cell that was studied in the skin. However, through ontogeny research, is now classified by and large as a macrophage with dendritic cell properties. So you can see how things get kind of murky. You have Kupffer cells in the liver. They live in the sinusoids. You have the alveolar macrophages that actually live in the alveolar extratissular space. And then we'll talk a little bit about cardiac macrophages, some more about the lamina propria in the gut macrophages, and then to a larger extent, microglia. These are all macrophages. So as I promised, we'll hear about Van Firth, because Van Firth's paper here in 1972 established the mononuclear phagocyte system, but put forth the model that these cells were derived from the bone marrow, from the adult bone marrow. So you have precursor cells in the bone marrow. You then have these promonocytes that give rise to monocytes that now can be detected in the blood, and these cells extravasate into the tissues and become your tissue macrophages. So here in connective tissues generally were called histiocytes, liver, as I mentioned before, Kupffer cells, long alveolar macrophage. Spleen, you had both free and fixed. Lymph nodes, same thing. Bone marrow with respect to the osteoclasts, serous cavity, I guess bone tissue here is osteoclasts, and the nervous system, microglial cells. These were considered part of the mononuclear phagocyte system, again, derived from circulating monocytes in the adult system, which were derived from the bone marrow. And just to show you, how recent, in 2003, relatively recent, the Van Firth model was still in play. There you can see this is a very important paper by Geisman and Stefan Young when they were in Dan Littman's lab, where they identified two different classes of monocytes. What you can see is the CX3CR1 high. CX3CR1 we'll be talking about more in a bit. This is the fractalkine receptor. It's a chemokine. It's highly expressed in monocytes and some macrophages, but what you'll see is CX3CR1 is expressed early on in the ontogeny, or the development of these cells, and then it gets turned off in some populations and maintained or even upregulated in others. But the point here is that you have two different classes. Here's the CX3CR1 high. This is now called the non-classical monocyte. And then you have the CCR2 positive, CX3CR1 low. This is considered now the classical monocyte. And as you can see here, the thinking was that these cells actually enter and become the resident macrophages in the tissues. So there was still relative adherence to the Van Firth model in the early 2000s. But it's important to note that there have been anomalies along the way with the Van Firth model in this period. You have, here are some examples. So for Langerhans cells, Hashimoto and Tarnowski here had noticed self-perpetuating Langerhans cells, meaning self-renewing. In other words, if these were always coming from the blood, then why are these cells then locally self-renewing? Here's another one: remarkable longevity. Again, if there's a continual turnover from the blood, then why are these seemingly long-lived? cycling population, self-renew throughout life. All of these sort of represented anomalies to the Van Firth model. You can see the same thing for alveolar macrophages. Tarling et al. In 1987 showed that there was self-renewal, Kupffer cells, proliferation. And then this one sort of hit the bank right before the major, I don't want to use the word paradigm shift, but change in the way we think about these macrophages in 2007, noting self-renewal in microglia. And that brings me to this next slide, this seminal paper by Ginou et al. Really changed, really was the tipping point in changing the way we think about macrophages. What they showed here is that microglia are derived from yolk sac macrophages. And the suggestion is that they self-renew and maintain themselves for life in this niche. In other words, they're independent from the pool of circulating monocytes in the steady state. So what they did was to use this RUNX1 Cree, Cree, with a Rosa YFP Phlox reporter. RUNX1 is a transcription factor. It was shown in this paper to be expressed in the yolk sac, in these blood islands, which were then shown to contribute to primitive hematopoiesis. So what one does is to pulse the pregnant dam at whatever embryonic time one wants to study. So in this case, they were looking at E7.5. And once you pulse with tamoxifen, that allows the Cree to enter and based off of the RUNX1 expression to now Phlox come in and enter the nucleus and Phlox out the stop codon and then allow for YFP expression. So it's driven by RUNX1. It's conditionally activated through tamoxifen, and then it maintains the YFP expression for the lifetime of the cells. And so what they did is they labeled these cells with 4-OHT. I think this is just the derivative of tamoxifen at E7.25, between E7.25, which of course you can do this reliably by counting plugs. And what they found, and this is your control, and what they found is that not only can you label these yolk sac macrophages from hitting these at E7.25, but these YFP positive cells can then be traced in the brain rudiment at a later time point. And in fact, that's what they're showing here. So when they pulsed these mice, you can see activation of being able to visualize these macrophages in the yolk sac, and then they move into the brain rudiment, where they showed this at a microscopic level as well. And the key to this figure here is that here they're looking at monocytes, microglia, and lung macrophages. This is the time point, 8 weeks, at which they're studying the pups from pulsed dams at the specific time point in which they pulse. And so this is essentially a pulse chase experiment. Here, they're pulsing at E6.5. This is with the RUNX1 Cree. And this is sort of before the hemogenic or blood endothelial RUNX1 expression takes place. So at 6 weeks, you don't see much by way of tracing these primitive macrophages into the brain. However, if you start here at 7.0 to 7.25, one can find these YFP positive microglia in the brain, which were pulsed again back at E7.0, E7.25, therefore effectively tracing these adult microglia back to their origins at the yolk sac. You can see the same thing at E7.25 when you pulse the pregnant dam. However, there's an important change here. Once you get to E7.5, E7.75, and further out, you now are hitting either the tail end of that RUNX1 wave, and you're sort of missing that wave. So at 8 weeks, you see relatively low recombination, and that even gets smaller. Now, what's now interesting though is you can see, and I'll talk a little bit about this, but the main point is that, again, pulsing at E7.0 to E7.5 allows you to tag microglia, trace microglia without other macrophages or monocytes. The other point, again, the smaller point I want to make is that you can see monocytes are being tagged here also. And that is eventually, once you miss that wave, you're now hitting into subsequent waves, which we'll talk about, that may hit the HSC, the hematopoietic stem cells, later in adult hematopoiesis, precursors for adult hematopoiesis. And so many papers followed this. Here's a critical paper by Frederick Geisman, Schultz et al. This was published in Science in 2012, essentially 2 years later. And they went on to basically show or say that, hey, wait a minute, microglia are not the only long-lived macrophages. In fact, you could look at most major organs and find evidence of long-lived macrophage presence. Now, I'll say that this is, as immunologists, we're probably not, as at the time anyways, very sophisticated with non-lymphoid tissue biology in the context of histology and analysis of tissue function. We basically just take the whole organ and grind it up and look for these cells by flow cytometry. And the case has changed. We now know that there's subtissular niches as well within an organ. For example, there are macrophages in the epidermis that are Langerhans cells, whereas the dermis has non-Langerhans cells type macrophages. But going back to just the points that this paper is making, again, is that you have these long-lived macrophages, and they happen to be MyB independent. MyB is a transcription factor involved in adult hematopoiesis. So you would expect that in the absence of MyB, you wouldn't get hematopoiesis at all. And what they show here is at E16.5 and later in these knockouts, you still have this population of macrophages that are maintained, albeit there's another population that seems to be MyB dependent. This is a CD11B high, F480 low, which is distinct from the F480 high, CD11B low. Okay. And what they then also showed in this paper is that you can take CSF1R, MER. CSF1R is, or CD115, is the colony stimulating factor 1 receptor. This signaling is critical for macrophage survival as well as differentiation. And you'll see that it's involved in the ontogeny or development. But so they took these mice at E8.5 and pulsed them with tamoxifen, same sort of concept here. So you're floxing the YFP in a tamoxifen conditional fashion or inducible fashion. This is the blood, and when they looked at the blood, you can see even in the Cree pulse mice, there's no YFP cells. And of course, the blood heart possesses the monocytes, and these monocytes are derived from hematopoiesis, adult hematopoiesis. By contrast, if we looked at when they looked at the liver, you can see a nice population of YFP positive, and this was Cree dependent, tamoxifen dependent of Cree, whereas the no Cree didn't show any YFP. And if you look closely at the populations within these tissues, remember the F480 CD11B population, F480 high CD11B population, you can see both populations in most of these tissues. And the only one that was YFP positive in the context of Cree are these, again, F480 positive or F480 high CD11B low. I wouldn't put too much stock in F480 and CD11B. I don't think that's really maintained too much consensus. But I think the underlying point is that when you pulse at E8.5, you do indeed see in the adult mouse, this is at 6 weeks, the presence of macrophages in most of these tissues. Okay. So following that paper, and we're now in 2015, there was another paper by Genu et al., Genu's group, saying, wait a minute, we agree based on our data that there are long-lived macrophages in each tissue, and this is not restricted only to microglia. However, there seems to be a difference in our findings, reflecting that there's actually two waves. There's an early EMP wave and a late EMP wave. The early EMP wave gives rise to microglia, whereas the later EYFP wave give rise to these other resident macrophages. And in addition to that, these other fetal these other macrophages, these non-microglial macrophages, go through a fetal monocyte intermediate that is derived from the liver. So let's just go through the data here. So here's the CSF1R, the same one, or similar one than the Geisman group used, and they pulsed at E8.5. And what you can see is, if you look at these tissues for percent recombination to show YFP, the only one at these different time points out to 6 weeks that has significant long-term expression of YFP are the microglia. This is brain. So liver macrophages, skin macrophages, kidney and lung all seem to lose the number of YFP cells, suggesting that it's being replaced by later waves. By the same token, using the Runx1 Cree, which we just talked about in the earlier Genu, has the same phenomenon here. So what you can argue is that these two cells that we're tracing here come from obviously the same origin. But by stark contrast, if you use the Runx1 Cree and go to E8.5, you see a flip. Now all the microglia are not labeled substantially, whereas all the other macrophage types are. So you essentially are hitting a second wave that occurs substantially at E8.5 that are now tagging these macrophages in other tissues, but not the microglia. And what they found was that this tagging existed in all of these tissues as monocytes as well. So you can see clearly that though there are macrophages being tagged, there are also monocytes in these tissues being tagged as well. And what we understand from monocyte macrophage biology, this is back from the Van Firth model, is that monocytes are the intermediate of early intermediate of macrophages. So this suggested to them, or at least clued them in the direction that perhaps these monocytes were giving rise to the macrophages in all of the other tissues, as opposed to, with an exception to microglia. And just to prove this, what they did was essentially do a transcriptome analysis of monocytes versus macrophages. And what they found was one gene, well, there were several genes, but one that they followed up on. And I don't want to put too much significance on the gene itself, other than to say that it was used as a tool to mark the two different populations. So it's expressed in monocytes, but not macrophages. And they showed that here by relative expression and then through qPCR. So so then they come in and use this S100A Cree, knowing that you're labeling only monocytes. And what you can see now is they proved that by labeling monocytes, you can also see labeling in macrophages in these tissues, again, allowing them to conclude that these macrophages, with an exception for microglia, come from monocytes. And they also went on to prove that these initially have were or originated one step earlier in the fetal liver. And I'm gonna sort of summarize this now. So now we have two models, and I can tell you that there's still a lot of controversy, but one model has sort of maintained more consensus than the other, and that is model 2, the Genu model. So let's just walk through this. And if you want Geismann's perspective, I've put this reference here if you're interested. And but so let's just go through these different models. So in the first model, we have model 1, we have the Geismann model. And in the Geismann model, it suggests that essentially you're hitting EMPs, and these EMPs seed the brain as macrophages. So they actually leave the yolk sac as macrophages. They don't go through a monocyte intermediate. And so they seed the brain, these are microglia, and then they seed all of the other tissues, like we said, the skin, etc., the lung, the liver. And so really there's no, based on his model, there's no distinction from the two ontogeny, which we know can't be correct based on Genu's study and other studies. There has to be a difference, and that is probably best explained here in model 2, although there's still open questions. Model 2 represents the Genu model. Okay, so you have these early yolk sac blood islands, and then you have at E8.5 the hemogenic endothelium, and these discern the two lineages. So in the early, this is CSF1R dependent erythromyeloid progenitor cells, they leave, or they're made in the yolk sac as a macrophage, and then go to the brain, and they get locked in. How do they get locked in? They get locked in because it's a closed niche. If you think about the blood-brain barrier, at that point, it's already established several days before. So they're really locked in. And the other tissues have more of an open niche to some extent, where in 8.5, while the macrophages that are made in the yolk sac from this EMP, and this is considered late MIB positive EMP, leaves the yolk sac, enters the fetal liver, and we know that the fetal liver is an intermediate hematopoietic organ prior to when the bones are fully formed and the HSCs are established in the AGM, and then finally in the bone marrow. So the concept here is these EMPs go in as monocytes, or they go in as progenitors, then become monocytes in the tissue, and then they begin to seed in the fetal liver, and then they begin to seed all of the developing tissues at that point, liver, lung, skin, etc., and maintain themselves throughout life. And I'll come back to that. And this third model, I don't really want to get into too much because it's relatively disproven, I would say, at this point. This was from a Japanese group who showed that in their system, and I don't remember their system committed to memory at this point, but essentially that the HSCs in the AGM is what contributes to the macrophages in all the tissues. And this can't be the case in terms of the adult system, as we had shown in some of the experiments prior. The one thing they do make the distinction clearly is that, the brain is different than the rest of the tissues. So at least that's consistent with the Genu model. But let us move forward, accepting this model as probably the best, closest to truth, quote unquote, and move forward from there, because I think that's what the field has set forth. Yes, I think we have a question.
Yeah, one quick question. So you mentioned that the microglia get locked in the brain by the blood-brain barrier. So does that imply that for diseases where the blood-brain barrier comes down, those microglia are able to move out of the brain, or do they still stay there?
The question is, do microglia leave the brain? Is that right?
If the blood-brain barrier is in some way compromised, because you
Oh,
mentioned
if it's
locked in
compromised.
by the blood-brain barrier. So that implies that it can.
There's some evidence that it can. But there's one paper I remember in PLOS Biology suggesting that microglia can leave. There's no reason for me to think why they would leave unless perhaps they wanted to get into the systemic circulation to somehow prime a T cell. So there's no real reason for them to want to leave. The general thinking about entry is cells enter from the blood. They don't necessarily leave from the blood. That's not to say that's absolute. They typically leave through lymphatics, and in the case of the brain, we have no lymphatics. In the case of the brain parenchyma, okay. Let me finish a couple more, either this slide or the next, and then just give you more opportunity to ask questions. So let me just give you this picture here where now we can differentiate the tissues from what we know. This was from 2016 by Ginou and Guillaume. This is a very popular, highly cited review. In what they're showing here, demonstrating, is in the brain, you have a yolk sac derived macrophage, and this seeds and maintains itself throughout life. Whereas the fetal liver monocyte derived macrophages, bone marrow, you don't see in this tissue. Now, this is in contrast strikingly to the remainder of the tissue. So the epidermis, which is the Langerhans cells, you do see an initial seeding of yolk sac derived macrophages, but eventually gives way to fetal derived monocytes that take up residence and maintain themselves. But what's interesting also to note is that the epidermis does have a small population of yolk sac derived macrophages, which and so this is established, but whether there's functional distinctions, we don't know in any manner of consensus, let's say. The lung and liver are quite different in that now they have fetal liver monocytes that make themselves into macrophages and maintain themselves, right? This is different than the epidermis, where there's still some mixing. And then you have these other tissues, which are strikingly different than the remainder, in that, yes, you do have yolk sac macrophage seeding. Yes, you do have fetal monocyte macrophage seeding and maintenance for a period of time, but eventually over aging, they get replaced from circulating monocyte or adult monocyte derived macrophages. And in the case of heart and pancreas, this is rather slow. So you can see the mouse is really aging before the monocyte derived macrophages really get a foothold. By the way, these monocyte derived macrophages are not maintaining themselves. It's a constant turnover, whereas these populations are maintaining themselves, for example, the fetal liver monocytes. And finally, you have the dermis and gut, which is a fast open niche. You can see within 2 months, the monocyte-derived macrophage adult hematopoietic really have gained the foothold and are the main dominant population. And the thinking for why this is unique in the gut is because in the gut you have microbiota and constant inflammatory triggers to some extent, peri-inflammatory triggers, which maintains constant turnover of the monocyte population. So let's just quickly revisit the Van Firth model. It's not that it's completely true. There are now major revisions to it. So it is true that most macrophages have a monocyte intermediate. So in most tissues, as I mentioned, even though they're long-lived, not even though they're long-lived, but they're long-lived in the lung, liver, skin, for example, those macrophages had a monocyte intermediate in the fetal liver in the prenatal period. Okay, so the monocytes are still the precursors there. The difference, the one difference is that clearly they're maintained. They're not coming from adult hematopoiesis. The other difference is, of course, the brain. In the brain, you have these yolk sac macrophages that are maintained themselves there for life. And then there's this other contingent group of macrophages that are actually derived from, you know, the original definition of Van Furth from circulating monocytes from in adult hematopoiesis, which I explained that exists in the gut and also in the dermis. And there are other examples as well. So I think I covered a lot of complex topics here. I wanted to take a brief moment and see if there's any questions before we move on. The crux of what I wanted to present to you has already been presented. So if there are any questions, feel free to please ask.
Looking at the three models that were presented, for the yolk sac macrophages shown in yellow in models 2 and 3, what becomes of them? They don't, at least on the
so
they appear to migrate anyway.
So, you know, so really what it is, think about it as being outpopulated. Eventually, new ones come in, and the new ones have a competitive advantage that maintain them. What the competitive advantage is, we don't fully understand. So the yolk sac macrophages might end up in the tissues, but eventually they give way to the fetal monocytes, which are just blasting and just basically taking over the niche and have a competitive advantage over the yolk sac. By this, you know, and you can make the same argument for the case of adult monocyte-derived macrophages in the context of the gut, which, you know, because there's a tonic low-grade inflammation, you constantly have monocytes being recruited, adult monocytes, and overcome that niche to a certain extent.
Gotcha. Okay, thanks.
Is it okay? That answers it. Okay. So let's move on. So one of the things that became evident was that ontogeny itself could not explain the full functionality of these tissue resident macrophages, because you see them in different tissues and clearly they have different functions. Even within, let's say, the fetal liver monocyte derived, if you look at, for example, the example I'll get into here is the alveolar macrophages. Clearly, those are distinct from, let's say, microglia, in that fetal macrophages are playing a role in surfactant homeostasis, which is unique to that tissue, whereas microglia are playing a role in, for example, pruning of synapses, which is in the CNS, which is quite unique to that tissue. And clearly, what they saw from transcriptomics was that there was some differences in the transcriptomes of these macrophages. So it suggested that the tissue itself had a large role to play in the function of the macrophage. And that's really what it's about in the end of the day. So this was quite an elegant experiment, or sets of experiments or manuscript by Guillaume's lab asking this question. And the way he did it was he took CSFR2 knockout mice. I think it's CSFR2 or CSF2. I think it's CSFR2 knockout mice, which cannot develop lung alveolar macrophages because they need signaling through CSFR2 to maintain themselves. So what they did was instead to intranasally transfer CSFR2 sufficient macrophages from other sources. So one thing is, remember, these are fetal liver. So one thing they did was to take yolk sac macrophages and these CSFR2 knockout mice, also comparing, doing the transfer with fetal monocytes as a control, bone marrow adult monocytes. And all of these, when transferred, were actually able to take up the niche, maintain themselves without monocytic input, and importantly, function in surfactant homeostasis. One of the major roles, critical roles, is macrophages in eating and clearing surfactant. So there's turnover of surfactant. Of course, that's critical for oxygenation. So this is one example how macrophages are critical in tissue physiology. They also then took fully developed tissue macrophages and tried that, but that didn't seem to work. So both at the monocyte level, either from fetal liver or from bone marrow, as well as from yolk sac macrophages, there was enough plasticity for these cells to actually take up that niche. And the other important thing here is, remember, bone marrow monocytes as I showed you in the previous sets of examples, are not long-lived and self-renewed naturally. However, once you put them, if you put them in the lung, or this is intranasally for lung establishment, they then can become long-lived and self-renewing and independent of the monocyte. So that facet of resident tissues may be contributed by the tissue itself, not necessarily imprinted from ontogeny. This is another example of this sort of question asked, now this time in the brain. This was done by Bennett et al. What they did was to take CSF1R knockouts. These mice do not have microglia. CSF1R is absolutely required for microglial survival. And here they then transplanted or transferred CSF1R sufficient monocytes or macrophages, I believe it's monocytes actually, into the mouse, both intracranial, I believe, also, as well as IV. And it turned out that the same phenomenon was seen, in that they are able to seed the brain, these microglial-like. Now, functional analysis was not done in this paper like it was here, to my recollection. But one thing they do note, which discerns it from this paper, is that while the larger transcriptome was relatively similar between the microglia and these microglia-like cells, there were critical genes that were different. This MS4A7 was expressed, for example, and CLEC12 and ApoE. So they had noted that essentially, while the broad outlook of these macrophages looked relatively similar, there are critical differences. So, and it's possible they may, in this paper, they may have missed the same, although they do show the important thing, which is a functional analysis here, which wasn't done here. But the point is that I think both papers you can take, despite their interpretations, both papers suggest that these monocytes can take on many of the characterizations that the tissue does, that it does in the tissue by way of tissue imprinting. But still, there seems to be something that is remarkably critical about the ontogeny, and I don't think we fully know the answer to that yet. And it might be different in other tissues. I see that we only have 5 minutes left, and I want to leave time for questions. So I'm just going to kind of zip through this last part. This last part is about inflammation. So recognize that in steady state, you have what I just showed you, in these tissue resident macrophages. But then on top of that, if you have an inflammatory event, now you have monocytes leaving the bone marrow and entering into the tissues in a CCR2 dependent fashion. And so you can argue that there are now two different populations in the inflamed tissue. And I think we're just starting to really understand what these are, how these are functionally different in the inflamed tissue. There's not consensus that the monocyte-derived macrophages in inflammation actually take over the tissue niche, maybe to a certain extent in some, but certainly not in the brain. So one has to then consider what these different populations functionally mean. And the problem is at least in the brain, that these macrophages are phenotypically indistinguishable. So how do you distinguish them if their markers are really pretty much the same? And in a stroke of genius, I would say these three papers, interestingly, most of them have overlapping mentors, came up with a beautiful answer. And this is the best way we can now distinguish, at least in the brain, microglia versus monocyte-derived cells using the CX3CR1 CRE-R and then a flox system. And that is because, as I mentioned, this fractalkine receptor is expressed both on circulating monocytes and microglia. However, circulating monocytes have a relatively short half-life. They live for 2-3 days, maybe a week and a half at most. So the floxed out reporter positive monocyte after this pulse of tamoxifen eventually gets washed out and replaced with fresh new precursors that are no longer tagged. Whereas in microglia, because they're long-lived, they maintain the floxed allele for their lifetimes. And in fact, in self-renewing, the daughter cells will carry this floxed allele as well. So you can now beautifully distinguish the two. And just to show you, we published this in Immunity in the retina. In the top half, you can see this is the retina without getting into specific details, but you can see microglia tagged with this TD tomato overlapping the CX3YFP expression. And now in an inflammatory setting, which we use toxic light, where you get degeneration of photoreceptors, you can actually discern the fate mapped or lineage traced microglia from the recruited monocyte-derived macrophages. And it turns out they are functionally distinct, but I won't get into that. So we have a few minutes left just to quickly overview the Van Firth model we talked about. And of course, the revisions to this model with identification of prenatal derived macrophages that seem to be that are independent of monocyte pool input. And we talked a little bit about ontogeny and tissue microenvironment or tissue environment in shaping macrophage identity. That is the future of this field. We're learning a lot about that. And also the future of the field is understanding steady state versus inflammatory macrophage input, which I just touched on. So we have a few minutes for questions. If there are any questions, please feel free to ask. I know this is a pretty complex topic, but hopefully I did it some justice. Seems like either everybody's so confused or they're not. I don't know. Hey, Carl.
Hey, this is your last lecture for the course?
I have one more on Wednesday.
Okay. Yeah, I may email questions later. I need this to soak in for a bit first.
Yes. Yes, it's very complex. All these changes have happened in the last 10 years or so. And you probably, I don't know if you'll even see it in textbooks at this point because it's so fresh. Like I said, in 2010 was that first major, was when the tipping point happened essentially. But yes, no problem. If you have questions for later on, please feel free. And in the next lecture on Wednesday, I'll be talking about dendritic cell ontogeny. I think that will also help to some extent because you'll see a distinction between dendritic cells and macrophages, which will help sort of solidify some of the information I explained to you today. Okay. Well, without any further ado, thank you guys. I'll see you on Wednesday. I'll be around. I'll stick around here if anybody has any questions they wanted to ask.
Thanks.
Shut off my, thanks, Carl. I'll just shut off the, turn off the recording.